Hi, and welcome to my OCR A-level biology revision session with me, Christine. So today's lesson I want to look at plant response and tropism. So what is a tropism in a plant? Well, a tropism is a growth response that the plant does to the stimuli from the environment. Now, the tropism is actually normally that this stimuli has come from one direction. So that growth response is going to go either towards or away from that stimuli. Now, the stimuli from one direction we say is unidirectional. And there are two that you need to know. Geotropism, the response to gravity, and phototropism, the response to light. So let's start with phototropism then. Now, it can either be positive phototropism or negative phototropism. So if we are looking at our shoots, for example, they are going to grow towards light. So we call that positive phototropism. When we look at our roots, they are going to grow away from light. That would be negative phototropism. So we're going to focus on the shoots today, and we need to understand that there are photoreceptors that are going to detect light. Now, these photoreceptors are found in the tips of the shoots. And what they do is they produce a hormone called auxin. Now, auxin is going to cause cell elongation. If we have light, which is unidirectional, coming from one direction, what's going to happen is the photoreceptors will detect that light, the cells will start to produce the auxin, and the auxin will diffuse. Now, when the light is coming from one direction, what happens is the diffusion of the auxin actually moves to the shaded side of the shoot, and that causes growth to occur, this cell elongation, which causes the bending of the shoot towards the light. So how exactly does auxin do this? Well, to understand, we have to take a step back and remind ourselves about plant cells and the fact that they have cell walls made out of cellulose. We know that because of the 180-degree inversion of the beta-glucose molecules results in these crosslinks being formed in the cell wall. So this is reminding ourselves about Module 2 knowledge. Well, what auxin does is auxin actually causes the cells to increase the transport of hydrogen ions from the cytoplasm into the cell wall. What that's going to do is that's going to change the pH, and that's going to activate enzymes called expansins, which are going to be useful in breaking these crosslinks between these cellulose fibers. That makes the cell wall very flexible, so it increases the plasticity of the cell wall. What that then means is this cell is now able to take water in by osmosis. Now remember, the fact of the cellulose cell wall was about maintaining that turgidity, that turgor pressure. It can withstand to a point, and then it will stop the cell from taking in any more water, preventing it from bursting. Well, by making the cell wall more flexible, it means that the cell can now take in more water and it starts to elongate, it starts to get bigger, and that is obviously going to cause pressure and cause that bending. If one cell on one side is longer than another, that will cause this strain, which will cause the bending towards the light. Well, it's not just about the increase in water and because of that flexibility in the wall. What we also know is that the auxin actually alters gene expression. And because it alters gene expression, it actually promotes the cell growth, which therefore means it promotes the production of more proteins. So we know that this is a hormone that is produced and secreted, it's then transported, and then it acts on cells which are called its target cells. So how do we know this actually happens? Where's the evidence? And this is the thing that they will do in the exam. They will give you potentially an investigation with a set of results and expect you to draw a conclusion. So when you're investigating phototropism in shoots, how do we prove that the auxin is produced at the tip of the shoot? Well, the first thing that they can do is they can remove the tip. That's the control. If they remove the tip and the cells do not elongate and the shoot does not grow any longer, there is no chemical signal for the cells to grow. So that's where the control comes in. Just by removing the tip of the coleoptile allows us to see that the tip is vital. Now, once they've removed the tip, what they can do is they can place that tip onto an agar block. Now, if they place it on an agar block, that's going to allow for those auxins to diffuse into that agar block. So if they take an agar block which has no auxins in it and they place that on the coleoptile that has had its tip removed and nothing happens, that again proves that the chemical messenger is the auxin. So now what we do is we take this auxin and the diffusion into that agar block and we place it right in the middle, central to our coleoptile which has had its tip removed. And what we note is that the growth is upwards. So because the growth is continually upwards, that shows us that if the auxin diffuses both sides, it will result in this growth upwards towards the light. If we then take this agar block which has the auxins diffused in it and we offset it, if we place it to only one part of the coleoptile shoot, what we notice is the offset block actually causes curvature. Now, because of the way of that curvature, that proves that as the auxin was diffusing down through the shoot, what was happening, it was causing cell elongation at that point. So therefore, if we mimic the unidirectional side of light, if we see the bending towards the light, that indicates that the auxin had to be diffusing laterally away from the light to the shaded side. So this is a way in which Darwin and Boyson actually proved it by doing an investigation, and they will give you practical information, either asking you to design the experiment or asking you in how can we prove or how can we look at the results and come to a conclusion. Now, the other thing to note is obviously at the tip we have light. If we covered the tip with a material that didn't allow light across, that would have an effect. So doing this investigation in the dark would be a way of checking the effect of light on that auxin production from the tip. The other thing you could do as well is you could alter this where you use a material, instead of using an agar block, for example, you could use a mica block that would therefore not allow the diffusion of the auxins, and place that into position and see how that blocking affects the elongation of the cells. So they will throw a random practical at you, but expect you to understand how to apply your knowledge of where that curvature is happening, that growth is happening, therefore the auxin was diffusing to that area. So the other one you need to know is geotropism. So geotropism is about the root's response to gravity. So the roots should grow down towards gravity, positive geotropism, and the shoot should grow away from gravity. Well, in this case, with geotropism, actually the auxin accumulates on the lower side of the root tip, and it actually inhibits cell elongation. So at the roots, auxin works slightly differently. Now, they don't expect you to know this in detail. What they do expect you to understand is that it's about the concentration of auxin, and they will give you information in a stem of a question and expect you to pull it out. So how would we investigate this? Well, with geotropism, we normally investigate in the roots by placing germinated seedlings in a petri dish which is vertically positioned. What does that do? Well, that ensures that the roots are going to grow downwards. If we place that also in a dark cupboard, what we're doing is we're preventing the effects of phototropism. So we're controlling the investigation. If we then leave that, what we would see is the results after 2 days of growth show us that the roots will move in the growth, they'll respond to where gravity is. So if gravity is below, you can see the root on the left-hand side seedling has continued to grow down. The one that was positioned in a horizontal position has actually had that curvature to grow downwards, and the one where the root was actually positioned pointing upwards has curved back down again. And if we rotate the dish at a 90-degree interval, as the seedling continues to grow, we will see this change in that growth of the root so that it will re-establish that response to gravity. So if they're going to give you a question based on this, they'll either ask you to plan the investigation or they will expect you to come up with a conclusion based on a set of results that you have been given. So I hope you have liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, please do check out my revision platform, www.aiqchat.com.